bacteriology, color atlas of medical bacteriology, 3rd edition, Mays, Bizot, and Peterson, Wiley. Table of content: Staphylococcus, Strep, Enterococcus, Aerococcus, Cryoniform, Listeria, Bacillus, Nocardia, Escherichia, Klebsiella, Yersinia, Vibrio, Aemonas, Pseudomonas, Becholderia, Cyanobacter, Actinobacter, Legionnaria, Neisseria, Haemophilus, Anaerobe, Clostridium, Campylobacter, Helicobacter, Chlamydia, Mycoplasma, Rickettsia. Antimicrobial susceptibility system, molecular diagnosis of bacterial infection, total laboratory automation. Here we find the author, Mesa, Preso, a native of Spain, University of Madrid, PhD from University of Minnesota, Basel, bachelor degree and master degree, science degree in medical technologist, Hartford Hospital, Cassinia, MD degree from Metropolitan University, Elena, PhD in microbiology and immunology from Georgetown University, Staphylococcus, catalase positive cocci, the most important, Staphylococcus aureus, Staph aureus. Here we find the gram stain, Staphylococcus aureus, gram stain, Macrococcus luteus. Here Staphylococcus aureus on blood agar, golden pigment of Staphylococcus aureus, blood agar. Size variation of Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus legonesis on blood agar, Macrococcus plutis on blood agar. Catalase, coagulase. This coagulase positive and negative. Coagulase test. The slide coagulate test is a rapid assay that tests for clumping factor on the surface of the organism. The test is performed by emulsifying the organism to be identified in both saline, which serve as a control for auto agglutination left, rapid plasma on right. Agglutinin of the organism only in plasma is a positive result. Staph aureus, the positive result, negative result. Coagulase test, a common method used to distinguish Staph aureus from other Staphylococcus species. The tube coagulase test shown the Staph aureus is positive. Colonies of the isolate to be identified were emulsified in 0.5 ml of rapid plasma. The tube was incubated 35 degrees centigrade for 4 hours and tipped gently to look for clot formation. The tube on the left is negative. While the tube on the right is positive. Evidence by clot formation, clot formation, tube giving negative result. Here the latex test for identifying of Staphylococcus aureus. In the test, latex particles have been coated with antibodies that can recognize bound coagulase as well as immunoglobulin that will bind to protein A present on the surface of the most strain of Staph aureus. Staph epidermidis, which serve as negative control and isolate to be identified where emulsified viscoated latex bead. The islet shown here was identified as Staph aureus. Here, aminotol salt agar. Aminotol salt agar is a selective differential media used for the isolation and presumptive identification of Staph aureus. The high salt concentration inhibit the growth of many organisms that inhibit skin and mucosal membrane. The phenol red indicator incorporated into the medium detect acid production, yellow here, resulting from the fermentation of mannitol. Staph aureus right were inoculated on the agar and then were incubated overnight. Mannitol salt agar. Mannitol salt agar containing oxycillin. Mannitol salt agar with oxycillin can be used to screen for presence of MRSA in nasal specimen, since the 7.5 salt and 6 mg of oxycillin in the medium inhibit most other organisms that normally colonize the nares. MRSA turns the medium yellow as a result of the fermentation of mannitol. Picture here is a plate incubated with methicillin susceptible Staph aureus strain and the MRSA strain. The methicillin susceptible Staph aureus strain failed to grow, as with most in vitro testing for methicillin susceptibility. Oxycillin, not methicillin, is used because of its high stability. Here is spectra. MRSA show in chromonogenic media used to detect MRSA. Spectra thermoscientific. DNase plate to differentiate Staphylococcus aureus. Thermostable endonuclease activity. Ornithine decarboxylase test for identification of Staphylococcus legionnaires. New protein susceptibility. Staph saprophyticus can be differentiated from other clinically significant isolate by its resistance to the antibiotic novobiocin. Bacitracin susceptibility. The same procedure used to test for bacitracin susceptibility for Staphylococcus biogenes can be used to differentiate Staphylococci with the bacitracin resistant from micrococci, which are susceptible. Lysostaphine susceptibility. Several species of Staphylococcus are susceptible to endopeptidase lysostaphine, which leaves a glycine-rich pentapeptide that is essential for cross-bridging the cell wall. Here the modified oxidase test, the microdase test from Thermoscientific. Also find Staph identification system. Durham's tube commercial system can differentiate among several Staphylococcus species. Fish FISH for the differentiation of Staphylococcus aureus from other in blood culture. Fish is a 90-minute fluorescent in situ hybridization. Assay utilize fluorescence labeled peptide nucleic acid. It's performed directly from the blood culture plot that are positive for gram-positive cocci in clusters. Staphylococcus biogene, Streptococcus biogene, group beta hemolytic streptococci, Streptococcus agalactias, Streptococcus equimilis, Streptococcus pneumoniae, very dense group Streptococci. Here we can classify the species, grouping within groups, Streptococcus, other group. Here gram strain of Streptococcus pyogenes group. Here gram strain of very dense group. A gram strain of Streptococcus pneumoniae, gram strain of Streptococcus mutans, blood agar, beta hemolysis and gamma hemolysis on blood agar. A key characteristic commonly used in identifying Streptococcus species is the type of hemolysis produced on blood agar. The three types of hemolysis are gamma hemolysis, and alpha hemolysis, which appear as a green of the agar around the bacterial colony, and the beta hemolysis in which the red cells surrounding the colony are completely lysed, which result in clear zone around the colony. Here, the Streptococcus pyogenes on blood agar. Pyogene group A produced a large zone of beta hemolysis around a relatively small colony. This organism typically translucent and has appearance of a small water drop on the large zone of beta hemolysis. Colonies have defined smooth edge. Here, Streptococcus pyogenes and Streptococcus constellus or small colony group Streptococci. Here, Streptococcus agalactia in Streptococcus pneumoniae, growth enhancement of Streptococcus, 5% CO2. Growth enhancement of Streptococcus on blood agar. Selection of Streptococcus agalactia. Pregnant female are screened for presence of Strep agalactia at 36 to 37 weeks of gestation by obtaining a vaginal rectal swab plated on the blood agar and placed in the selective broth as shown, incubated overnight, and the enhancement broth is subcultured to blood agar. Here, Grand-agar for detection of Streptococcus agalactia from clinical specimen. Grand-agar with enrichment broth. Here, carrot broth for direct detection of Streptococcus agalactia. Carrot broth can be used to both enrich for the identification of the Strep agalactia group B. Here, Bacetracin susceptibility test for presumptive identification of Streptococcus pyogenes. Test is rapid sensitive test for identifying of Strep pyogenes. Here, CAM test for the identification of Streptococcus agalactia. Group B Streptococcus. A CAM test can be used to presumptive identifying of Strep agalactia. To perform the test, the beta-lysine producing Staphylococcus aureus strain is inoculated in thin line onto the blood agar. The isolate be identified is inoculated at the right angle to Staph aureus line. Take care that the two streaks don't touch. After overnight incubation, Strep agalactia, the area of beta hemolysis where the two organisms have shown in proximity to one another, should take the shape of an arrowhead of beta hemolysis. CAM test using beta-lysine disc. Epurate test for identification of Streptococcus agalactia. Here, Optocin susceptibility for identification of Streptococcus pneumoniae. Bile susceptibility test for identification of Streptococcus pneumoniae. The differentiation of Streptococcus bovis group from Enterococcus faecialum. STR rapid system for identification of Streptococci. The bacteriology Enterococcus. Members of the genus Enterococcus are gram-positive cocci, can survive harsh conditions, isolated from reduced susceptibility to vancomycin, can be catalyzed by resistance gene. Here gram stain of Enterococcus faecium, sorry, Enterococcus faecalis, Enterococcus classificator, vancomycin dependent Enterococcus faecalis strain. The biochemical test system: nucleic acid, Enterococcus. This is the classification of Enterococcus. Here the gram stain, Enterococcus on blood agar, the biochemical test of vancomycin resistance, chocolate agar, satellite growth, cranioform gram-positive bacilli, cranibacterium, its classification, gram-bacterium. Organisms include cranioform bacteria. Here we classify clinical source of type of infection and characteristic. Every type, the biochemical test, here gram stain of cranibacterium diphtheria, biochemical test, blood agar, gram stain, Listeria, Bacillus rex, it's blood agar, its isolation, camp test, Bacillus, slight gram stain, blood agar, biochemical test, Eucardia, gram stain, colonies, biochemical test, Mycobacterium, its classification, tuberculosis, bovis, Cabri, biochemical test. This is quinoion strain of Mycobacterium tuberculosis, sputum, Oramine stain, gram stain, Mycobacterium, quinoion and Oramine stain, Mycobacterium leprae, Mycobacterium tuberculosis, Levenstein Jensen media, Mycobacterium avium, Mycobacterium intracellularis, Mycobacterium Levenstein Jensen agar, Mycobacterium collini on Levenstein, biochemical test, growth of McConkie agar without cryptal violet, iron uptake test, niacin accumulation, biochemical test, enterobacteriaceae, grower rapid late, classification, gram stain of Enterobacteriaceae, Klebsiella pneumoniae, on blood agar, Proteus species, Yersinia intercolitica, Serratia species, Escherichia coli, Klebsiella, Escherichia coli and Shigella, Salmonella colonies, positive Salmonella colonies, Escherichia coli, Yersinia intercolitica, biochemical test, Escherichia, Shigella, Salmonella, gram stain, colonies of Escherichia coli on maconche, colonies of Escherichia coli on agar, colonies of Escherichia coli in coro magar, biochemical test, colonies of Shigella and Escherichia coli on maconche agar, Shigella species, TSI agar, Salmonella species, Salmonella serotype typhi, Salmonella XLT4, Salmonella enteric, Klebsiella, Enterobacter, Citrobacter, gram stain of sputum species, Klebsiella, Klebsiella ulmaconchi, Serratia, biochemical test, colony of Klebsiella pneumoniae, colony of Klebsiella pneumoniae, Proteus on blood agar, Odwarsiella, Hafnia on maconchi, Plesiomonas, Scheloides on maconchi, Yersinia, gram stain, Yersinia enterocolitica, Yersinia on blood agar, Yersinia on maconchi, biochemical test, Yersinia fish, Yersinia on blood agar, Vibrionice, Vibrio, cholera on blood agar, gram stain of Vibrio species, Vibrio on blood agar, Vibrio on colnera, Vibrio on TCBS, Vibrio on TCBS, Vibrio parahemolytic, Vibrio mimic, Eromonas, gram stain, Eromonas on blood agar, Eromonas on maconchi, Eromonas in ectonagar, Eromonas KV, Pseudomonas, gram stain of Pseudomonas aeruginosa in Umbeladagar, Pseudomonas aeruginosa in McConkey, microcolonies on Pseudomonas aeruginosa in triple sugar, Pseudomonas aeruginosa in molar Hinton agar, Pseudomonas Umbeladagar, biochemical test, Pseudomonas in fish,